Today we're talking about Chapter 7, Section 2, which is a continuation of our discussion of these non-Mendelian patterns of inheritance. And what we will focus on today is how the phenotype of an organism is affected by many different factors, not just simply whether you have a dominant or recessive allele. Our first example that we're talking about is called incomplete dominance. So think of that word incomplete. Neither allele is completely dominant, nor is it completely recessive. What happens is when you have two contrasting alleles, they kind of blend together to make a third phenotype, which is some kind of intermediate. Our example that we have here is a flower called a snapdragon. You can see that we have a red flower, which is the result of two red alleles, or a white flower, which is the result of two white alleles. When they are crossed, they make a third phenotype. So one red allele and one white allele together make a pink phenotype, which is kind of blending the two traits of the parents. So it actually turns out that even though Mendel disproved the blending hypothesis of genetics, it is true for some genes in some organisms, and that's exactly what we see here. So let's talk about how to make a Punnett square for incomplete dominance. You'll notice that we have two different letters. We're not using a capital and a lowercase r because we don't have a true dominant and recessive allele. So we just use two different letters, R for red, W for white, and then if we have RW, we know that we must interpret that as a pink phenotype. So if you make your Punnett square, what you will see, regular 4 boxes, we put the two R alleles from one parent across the top, we put the two W alleles down the side, and all of our offspring we would expect to be heterozygous, one R and one W, and that gives us a phenotype of pink flowers. Related to this is also an exception to the law of dominance. We have a condition called codominance. This happens when both alleles are completely expressed. Co means together, so they're kind of dominant together. One is not dominant over the other, they would both be dominant at the same time. And neither one of them is going to be truly recessive to the other one either. They can both kind of just coexist. Our example is this color pattern that we call roan. You can see it in the offspring right here. It's kind of this spotted pattern. And if we take a bull that has red hair, and even though it might look brown to you, it does have a reddish tint. This would typically be called a red coat color. And if we cross him with a cow that has white coat color, they will produce offspring that are this kind of spotted roan coloration. So there's some patches where that red color is completely expressed, and then there's other patches where the white color from the mother is completely expressed. So red doesn't cover up with the white, it doesn't blend with the white. They're both fully expressed at the same time, just in different regions of the body. If we make a Punnett square, it's going to look very similar to what we just made in incomplete dominance. We're still using two different letters because we don't have one thing being dominant over the other. And in that Punnett square, we cross that red bull with the white cow. We can see 100% of the offspring would be RW for their genotype, and that would be a roan color. So again, codominance would occur anytime you still see both of those parental phenotypes in different parts of the body of the offspring, whereas incomplete dominance, they were blended together in the offspring. So that's how you can tell the difference between those two. Our next exception to Mendelian inheritance is called multiple alleles. This would mean that there are more than two alleles for one gene. When Mendel studied his pea plant characteristics, each characteristic only had two alleles. For example, there was a purple flower allele or a white flower allele. Or if he's talking about the seed shape, there was a round allele and there was a wrinkled allele. And that's true for a lot of different genes, but we are increasingly finding more genes that have multiple alleles, meaning more than two versions of that gene. Now, even though there's more than two alleles possible, a diploid individual, which we all are, can still only inherit two out of the possible however many alleles there are. So that's important to know. Even if there's four alleles possible for a gene, you as an individual can only inherit two of them at a time. And the classic example of multiple alleles in humans is the blood type. So we've got on the blood cells here, a little background information. Blood cells have these proteins that stick out. And these proteins are different shapes on the end, and that allows the blood cells to be recognized by your body. There's one shape that scientists have just called shape A, so that would be antigen A. There's a different shape, which is called antigen B. And then it's possible to have both of them present on your cells at the same time, or it's possible to have none of them present on your cells. So these different antigens here are where the name of your blood type comes from. There is a type A blood, which means your blood cells will only have antigen of type A. And the genotype for that, you'll see the genotype written with the letter I. The letter I just refers to the gene that produces that antigen. That gene I, if it is type A, we use a superscript of an A. And you'll notice this lowercase I here. Well, that's the recessive form of having no antigens whatsoever. So there's two genotypes that would give us type A blood. You could either be homozygous and have two type A alleles, or you could have a type A allele and this recessive lowercase I, which would just be a type O allele. Same for type B. You could have two B alleles, or you could have a B and that recessive O. If you're type AB, however, you have to have one A allele and one B allele. So there's only one genotype that would give you this phenotype of type AB blood. And finally, if you are type O, there's only one way you can be type O, and that's if both of your I alleles are this lowercase type O allele, and then your blood cells would not have any antigens on them at all. So taking all of this information into account, we have a question here that's a good chance to practice. What is the likelihood that a man who is heterozygous for type B? Well, if he's heterozygous for type B, that would be the second genotype here, meaning he has one B allele and his other allele is an O. And then we're looking for a woman with type AB blood. Well, there's only one genotype that she could be. She has to have an A allele and a B allele. So if we make this Punnett square, here is the man who is heterozygous for type B. Here's the woman with type AB blood. If you fill in those boxes, which we fill in just like any other Punnett square, you can see that this child here would be type AB. This would be a type A. This would be type B blood with two B alleles. And this would be type B blood, one B allele and one O allele. So the answer to our question, the likelihood of them producing a child with type B, is going to be 50%. Now our next example is similar to multiple alleles, but in this case, we are talking about polygenic traits, meaning there are multiple genes. So not just one gene with several different alleles, but separate genes located on separate parts or different areas of the chromosome. So this is different from what we just discussed. Multiple genes or polygenic inheritance is going to be what gives us all of these traits that have a really wide variety. Like eye color is actually way more complex than just do you have blue eyes or do you have brown eyes. There are at least 3 different genes, and for each gene, they each control a portion of what your eye color is. So you could have a brown or blue allele for this BEY1 or a brown or blue allele for BEY2. So depending on which combination of alleles you get, you can have any one of those eye color combinations, or even some that aren't pictured here. The good thing about a polygenic trait is that you're never going to have to draw this Punnett square. This is a pretty simple version of a polygenic trait for skin color. It only shows 3 different genes. In reality, there's probably way more genes that have control over what skin color you have. But even in this simple version, look at all of the different skin colors possible for just 3 gene locations. And basically, if you were to put them in a graph, notice how it makes a bell-shaped curve. So the thing to know about polygenic traits is not how to make a Punnett square for them, but to recognize that if you have a very wide range of phenotypes, that phenotype is almost definitely controlled by multiple genes. The only way you can have all of these different phenotypes possible is if there are multiple places where that phenotype is controlled, meaning multiple places on your DNA, multiple genes that can be expressed or not to give you your final phenotype. Height would be another one that would be a really good example. There's almost an infinite number of different heights that humans can be, because you don't just have to be 5'6" or 5'7". You can be any height in between, even down to just the fraction of the inch. So again, a polygenic trait, the thing to recognize is when you have a very wide variety of different phenotypes, that has to be controlled by multiple genes. Our next example is a really cool example called epistasis. And this also involves multiple genes. And this is when one gene literally controls another gene. So we have 2 genes at play in this example here. You've probably seen Labrador retrievers. You know they have different colors. They can have black fur, or they could be brown, or they could be a light color like a yellow lab. So the genes that control the coat color, there's one gene, gene B, that controls the color of the pigment in the hair. And black would be dominant over brown. So if a dog has 2 lowercase b alleles, the pigment color they produce is brown. But if they have at least 1 dominant black allele, then they will produce black pigment. But you may be wondering, where does this yellow lab come in? Well, this comes in if you look at gene E, and this is the epistasis, so that's why it's called E with the letter E. You'll notice that in this yellow lab, there are 2 recessive E alleles. That means that there is no expression of color at all. It does not matter what this yellow lab has at gene B. It could have 2 capital B alleles and still be a yellow lab because it's recessive at this second gene location. In other words, the only way to get either brown or black color is to have at least 1 dominant allele for gene E. Having a dominant allele at gene E basically says, yes, gene B, go ahead and express whatever color you have the code for. But if you are recessive at gene E, there's no color expression at all, and that's why you have those really light-coded labs. Our next example of non-Mendelian inheritance actually has to do with more than just the genetics of the organism, but the impact of the environment on the genotype. So there's a lot of traits that definitely have a genetic basis, but are impacted by how the organism grows and develops in its particular environment. A good example of this would be the sex of sea turtles. When those eggs are laid in a nest, there are temperature variations within the nest, and the warmer areas of the nest are going to cause those eggs to develop as female, where the cooler areas are going to cause those eggs to develop as male. So there is still going to be a mix of male and female sea turtles, even from within the same nest, because we're talking about a pretty narrow temperature range here. This is only about 4 degrees Celsius from either extreme to the other. But that importance of the environment definitely impacts the development of the phenotype of the individual. This happens in humans as well, not for our sex determination, but for other things such as height. Your height definitely has a genetic component, but also it is impacted by what type of food you eat as you're growing up. Are you going to eat foods that keep you well nourished and able to grow at your optimal rate? Or if a person is malnourished as they're growing up, that's going to impact their height and how tall they grow. So the environment definitely plays an important role in the expression of some of our genes. And finally, our last example of these complex patterns of inheritance is called non-nuclear inheritance. Non-nuclear is exactly what it sounds like. This would be when a trait is inherited from DNA that is not inside of the nucleus. All of the genes that we've talked about so far are on the chromosomes in a eukaryotic cell that are housed in the nucleus. But remember, there are organelles in the cytoplasm that also contain their own DNA, namely those chloroplasts and mitochondria. The DNA inside both of those organelles mostly contains genes for how the chloroplast functions or how the mitochondria functions. And some of those genes, if they're mutated, they could have a visible phenotype or effect on the organism. One example of that would be in the fast plants that you see here. These are plants that have this variegated color pattern, which just means you see that variation of kind of stripy looking color pattern. This is inherited completely through the chloroplasts of the plant. Remember, chloroplasts have the chlorophyll in it, and so those chloroplasts that are passed down from mother to offspring, if they contain a gene for this variegated color, then one mother plant would produce all offspring that contain that gene. It's important to note that for non-nuclear inheritance, this is only coming from the organism's mother. Remember how we talked about in meiosis, that uneven division when female gametes are formed, and all of the cytoplasm and the organelles contained in the cytoplasm end up in that one egg cell. That's the only source of chloroplasts or mitochondria or all of the other organelles for that fertilized egg, which goes on to become the zygote. So all of your mitochondrial traits you inherited completely from your mother. The sperm cell does not contribute any mitochondria to that fertilized egg. So anytime we have non-nuclear inheritance, we are looking at examples of the female parent passing down those alleles for those genes, and there's no contribution whatsoever from the male parent. So that's going to be the end of our discussion about complex patterns of inheritance. We are going to be doing plenty of practice with this in class. So if it doesn't quite make sense to you yet, then make sure you take advantage of that practice. And as always, please make sure you ask questions if there was anything that you find difficult.